Hello, everyone. It is my pleasure to introduce you to this webinar on finding evidence in biomedical publications. My name is Maria Levchenko. I'm part of the team working at the Europe PMC literature database at the European Bioinformatics Institute. Europe PMC provides many useful tools to assist you with your research. One of our key features is literature data integration. In this webinar, I will show you how you can track primary research data in scientific publications using Europe PMC, and how you can take advantage of the text mining tools that we offer to find facts and evidence in research papers to support or refine your own hypothesis. All of the resources that we will talk about today are open, free, and easy to use. They do not require a bioinformatics background or programming skills. So what is Europe PMC? It is a free digital repository of biomedical and life sciences literature. We are based at the European Bioinformatics Institute, part of the European Molecular Biology Laboratory in Hingston, UK. Europe PMC is supported by 28 international research funders. We are the designated repository for researchers funded by these organizations to meet their open access publication mandates. You can access the resource at europepmc.org. Europe PMC was set up in 2007 as a partner in the PubMed Central International Initiative. Together with PMC USA and PMC Canada, it constitutes a network of digital archives for published peer-reviewed biomedical literature. All three organizations share the full-text publications. Europe PMC, however, provides access to additional literature sources and other services and features on top of the content. Europe PMC combines almost 4.5 million full-text articles and nearly 33 million abstracts, mainly from PubMed, but including other sources, as well as patents, theses, clinical guidelines, and grant information. Importantly, Europe PMC operates a unified search. When you look up a keyword in PubMed or PubMed Central, only abstracts or full-text articles are included in your search. Europe PMC is a single resource, combining abstracts from PubMed and the corresponding full-text article from PubMed Central. It saves you the trouble of searching both databases and expands your search. Europe PMC is an extensive resource, and it continues to grow. More than 2.5 million scholarly articles are published every year. Even when focusing on a single topic, it can be hard to stay on top of your field. Imagine working on chronic kidney disease. There are over 400,000 papers on this condition to start with, and each year the number increases by 30,000. That's a lot of papers to read. It is not possible for an individual researcher to process the vast amount of information in these papers. Moreover, the papers themselves become more complex. A single publication may be packed with a whole variety of research data. This means that you often have to operate several resources at the same time, comparing sequence variants from healthy and diseased individuals, while also looking at the protein structure to see how mutations would influence protein folding, checking with a small molecule database to see if ligand binding would be disrupted, and perhaps scrolling through a review describing the disease symptoms. It becomes harder to integrate these diverse data formats into a coherent workflow. To help you navigate the data-rich literature in the most efficient way, we at Europe PMC have created Scilite. Scilite is a tool that highlights biological terms or relations identified by text mining on abstracts and full-text articles. It helps you to read the paper faster, focusing on important concepts, and to find data and evidence within the publication. Let's see how it works. When reading a paper, on your right-hand side, you will notice a menu titled Show Annotations in this abstract. By checking the corresponding boxes, you can select different concepts that you would like to be highlighted in the text. You can also uncheck the boxes to remove highlights from the text. Among annotations that you can select are biological entities, such as gene and protein names, organisms that correspond to species names, chemicals, diseases, and gene ontology terms. For those unfamiliar with gene ontology, it is a controlled vocabulary that defines concepts describing gene function. Membrane, transcription, catalysis, those are some examples of gene ontology or GO terms. You can also find annotations for protein phosphorylation, gene function, as well as relationship information such as gene-disease associations and protein-protein interactions. As I have already mentioned, the annotated concepts are identified by text mining algorithms. Some of those are developed by the Europe PMC team, and others are submitted by our collaborators. For example, gene-disease relations are contributed by two groups, Open Targets and DisGenet. Unlike other annotations, protein-protein interactions are not derived by text mining. Those annotations are manually curated by experienced researchers at Intact Database. Scilite is an open platform for annotations. Other research groups can submit their results using the Skim. This means that the number of annotations is set to grow in the future, highlighting further kinds of concepts and relations in the text. So how can you use Scilite to help you with your research? If you need to quickly scan the article for keywords to understand if it is relevant to your topic, or if you would like to skim read the paper, focusing on the important bits without having to read the entire article, you can simply switch on the highlights for annotated concepts. In the two examples here, you can see that the excerpt from the first paper mainly discusses two proteins, RARE1 and NED4, in human and yeast. In the second paper, researchers have studied the following tardigrade species, highlighted in yellow. Scilite can help you see what are the main players described in the text, which model organism the work was done in, or the kind of drugs that were used for treatment. Another Scilite function that can be useful for skim reading is providing a list of terms found most frequently in the text. To get to the list, click on the right arrow next to the annotation type. A selection of individual terms will appear together with up-down navigation buttons which allow you to jump to selected terms in the text. You will notice that some of the terms could be listed several times. For example, mycobacteria, Mycobacterium tuberculosis, and M. tuberculosis may be referring to the same organism. However, the text mining algorithm will list them as separate entities. Nonetheless, the list can give you an idea of the most common concepts appearing in the text. To get back to the annotation types, click on the left arrow, and this will bring you to the familiar menu. Notice the number in brackets next to the annotation name. It indicates how many different terms belonging to that group were found in the text. Although text mining algorithms are getting better and better at getting the most meaningful and useful information, it can be hard to discern terms due to similar names or abbreviations. For example, peanut can refer both to plant species and Drosophila gene name. Or, in the example here, acronym APC can correspond to one of the following concepts: antigen-presenting cells, activated protein C, anaphase-promoting complex, or argon plasma coagulation. Sometimes, despite stringent quality checks, you may notice an annotation that is too generic or incorrect. It is of critical importance that the readers find the annotations useful, so you can provide feedback on each annotation. To do so, use the feedback link in the pop-up window. You can also endorse annotations using the feedback link if they're useful to you. This information is fed back to the Europe PMC team and will be acted upon, helping to improve the annotations overall. While reading a publication, and depending on your background, you may find some unknown concepts in the paper, or perhaps you might need some additional information about a familiar concept. The beauty of Scilite is that annotations are linked to structured resources in public life sciences and chemistry databases. If you click on one of the highlighted terms, it will bring up a pop-up menu with a link to associated data record. For example, when clicking on methoxymucolic acid, a chemical molecule, you will see a link to CHEBI, a dictionary for chemical entities of biological interest. If you follow that link, you will see a summary dedicated to this particular compound. You can find the chemical formula, mass, and charge, as well as some functional information. Similarly, if you open a pop-up for genes and proteins, you will see a link to UniProt, a knowledge base of protein sequence and functional information. These links from text to structured resources create a knowledge map of what is known about biological objects. If you're exploring a new topic, such links provide you with encyclopedic coverage that comes from reliable public data sources. You may not be familiar with the resources commonly used in the field, so you can rely on Scilite to guide you to a trusted, high-quality database. Moreover, Scilite can help you find evidence and formulate or refine your hypothesis. As I have already mentioned, Scilite annotations include functional relations, such as gene-disease associations and protein-protein interactions. For those annotation types, an entire sentence that contains the evidence for this relation is highlighted. Here is one example highlighted in a shade of red. In this case, we find the evidence that NKX2-5 protein interacts with TBX5 transcription factor according to a yeast 2 hybrid experiment. If you click on the annotation, you will again see the pop-up menu. On the menu, you can see the link to the source with additional description of the interaction details and the annotation source. In this case, it is the intact database. The pop-up will also provide a brief statement on the evidence for this interaction. As I have mentioned, for this example, it's physical interaction confirmed by the yeast 2 hybrid assay. For gene-disease associations, you will find two links displayed on the pop-up menu, one for the gene and another for the condition associated with it. In some cases, a sentence will contain overlapping annotations. If that will be the case, Scilite will highlight the longest annotation, and the individual annotations within the phrase will appear in the pop-up window together. You can interchange between them by clicking on the annotation name in the pop-up, like shown here. Finally, there is one more function that Scilite can provide to support you in your research. It enables scientists to locate the primary data from the text. For instance, if you would like to retrieve and use a public data set to cross-reference a result or test your hypothesis in silico by using public omics data, you can focus on accession numbers in the text that link to that primary data. Accession numbers are unique identifiers given to a data record to track it in a single data repository. Such identifiers are crucial, especially because protein and gene names are often ambiguous. Accession numbers are yet another type of Scilite annotations. They are especially useful because they allow you to quickly get to the data mentioned in the paper, be it protein structure coordinates or sequencing results for biopsy samples. Here, in this example, you can find a reference for a PFAM record for a protein domain in the text. Scilite tracks accession numbers for several different resources, including ArrayExpress, UniProt, PRIDE, the Proteomics Identification Database, Protein Databank Europe, European Nucleotide Archive, and many more. You can see a full list on this slide. If you click on the highlighted accession number identified by Scilite, you will get an already familiar pop-up menu with a link to the corresponding data source, where you can view, download, or analyze this data set in question. It can be difficult to make sense of the raw data in its natural form, so we experiment with data visualization techniques to help you digest information efficiently. For instance, Scilite displays a 3D view of the protein structure on the pop-up menu for datasets deposited in Protein Data Bank, or PDBE. This acts as a preview to the actual dataset, allowing you to assess the structure in its original context and get a visual cue for a wordly description. You can then continue reading the paper or proceed to the database to work with the structural data. When using Scilite tool, you will notice that full text of some articles doesn't have an annotation menu apparent on the right-hand side. This is because the use of the material is subject to the copyright and related license terms of the respective authors or publishers. Scilite annotations can only appear on full-text articles with a CC BY, CC BY-NC, or CC0 license. However, we do text mining for the entire full-text corpus and display results in a separate tab titled Bioentities. On the abstract or full-text pages, you will see the Bioentities tab. If you click on the tab, you will be able to browse a list of concepts and entities that is similar to the list of Scilite annotations. Clicking on the name of a bioentity will take you to the corresponding database. Note that Bioentities tab and Scilite link to different resources. This is because the focus of the Bioentities tab is on individual text-mined terms rather than the primary database record. Clicking on the search icon next to the term initiates a search on Europe PMC for that term. You will also find links to the datasets mentioned within the article. Data citations in the Bioentities tab are based on accession numbers, or DOIs, which you have already encountered when using Scilite. In addition to the data cited in the publication, you can also see database records that have cited the article. When datasets are submitted or curated, references to the literature are frequently added, and those references are made available via the Bioentities tab as database citations. Both types of citations can be explored via the Advanced Search form. On the Advanced Search page, select Data Links and Data Citations. You can see all available options in the drop-down menu. This data search functionality distinguishes Europe PMC from other resources. Most databases will provide links to underlying primary literature, but these links might be scarce, let alone extensive. You can easily search for data associated with certain concepts or keywords and create complex data-driven search queries across multiple resources using Advanced Search from Europe PMC. For example, you can look up which publications on ulcers have a link to clinical trials, which articles on Arabidopsis thaliana provide associated mass spec data, or which papers provide both structural and functional information for protein domains implicated in unfolded protein response. Here's one example of a complex data search. Let's say we're interested to find papers that work with Haemophilus influenza as a model organism. So we search for this concept in the methods section of the article. We would like to limit our search only to publications that report mass spectrometry data deposited in proteomics change. So we select it as a data citation source. Now our search brings up two publications. Let's go to the first example. When we select the full text, we can highlight the accession number for the mass spec dataset. Here's the link to the record. Let's also check that the paper actually deals with the organism we have selected. Clicking on the organism's annotations should highlight the species in the paper. It takes a while to load all annotations. This is probably because there are many instances found in the text. Indeed, there are 276 mentions. If we now open the list, we can see Haemophilus influenza and go into the mentions. Now it shows us that it was indeed used as a model organism. Now you know how to search for and identify primary research data in scientific publications. However, there may be several different types of data from a variety of sources associated with a single paper. For instance, this publication is citing over 30 different data sets, including a protein structure from PDB-E, several genomic sequences provided by ENA, and an mRNA sequence from NCBI nucleotide database. In addition, it also has several supplemental data files. To explore all data associated with a particular paper, you can view its biostudies record. Biostudies is like a bento box for data that packages all data about a study in one location. You can access the link to Biostudies record from the abstract page of the paper. Within the database, you'll see links to supplemental files as well as links to cited data in this record together with the study abstract. With this, we have hopefully covered all different ways in which you can explore the connection between scientific literature and research data. For more information, you can refer to Europe PMC help pages. If you're interested in the mechanism behind Scilite application, you can follow the link on the slide to the software tool article. You can also view further training materials on Europe PMC that can be found via the following link on the EBI Train Online pages. If you ever have any questions in the future, please feel free to contact us via email. You can follow us on Twitter, and if you're interested in the use of our API, please join the Web Services Google group. You can also view upcoming EBI webinars at this link, and we'd welcome any feedback you might have. Thank you very much for your attention. It was a pleasure to deliver this webinar to you, and if you will have any further questions in the future, please direct them to the Europe PMC team. Thank you and goodbye.